At the surface, a scarlet shrimp burns against blue water like a moving ember. Carry that same animal downward, and its vivid armor seems to fade toward black. The transformation is not a change in pigment, but a change in available light. This disappearing act begins because seawater filters sunlight unevenly as depth increases. Long red wavelengths surrender first, absorbed within the ocean's bright upper layers. Orange and yellow weaken next, leaving a narrowing world dominated by blue-green photons. A red surface reflects red light, yet deep water supplies almost none to return. What once looked conspicuous, therefore, becomes a dark shape against a dim background. The mystery is not why red pigment stops working, but why darkness makes it useful. Many deep-sea crustaceans, fishes, and gelatinous animals carry red or reddish tissues. Their coloration survives where human eyes would expect pale bodies or simple transparency. To understand that pattern, you must follow light rather than color names. Each descending meter changes the illumination striking skin, shells, muscles, and internal organs. The ocean turns a familiar hue into camouflage through ordinary absorption and reflection. Our investigation descends with that shrinking spectrum, testing where the concealment succeeds and fails. Sunlight contains many wavelengths, which surfaces absorb, scatter, or reflect in different proportions. Objects appear colored only because some wavelengths reach them, and then reach an observer. Pure water absorbs longer visible wavelengths more strongly than the blue region of sunlight. Dissolved substances and suspended particles modify that filtering, especially in productive coastal seas. Consequently, no single depth marks the universal moment when every red animal becomes dark. Water clarity, solar angle, weather, and viewing distance all alter the remaining spectral field. In clear, open ocean, blue light penetrates farther than red light under comparable conditions. In turbid water, scattering and absorption can darken the scene much closer to the surface. Laboratory spectra confirm the physical trend, while underwater cameras reveal its visual consequence. A diver's white lamp restores missing wavelengths and suddenly resurrects crimson colors from apparent blackness. That dramatic return demonstrates illumination, not spontaneous biological color change behind the effect. Yet red camouflage is not identical to invisibility, because outlines and reflected blue light remain. Dark red surfaces can absorb much of the surviving blue-green illumination that strikes them. Some animals combine red pigmentation with body shapes that reduce recognizable edges. Others occupy midwater, where concealment must work against both horizontal gloom and downwelling light. Several mesopelagic shrimps appear intensely red under lamps, but extremely dark under ambient conditions. Their pigments can also mask luminous prey glowing inside translucent digestive tissues. That concealment matters because a bright meal could advertise a predator's position. The cue is internal bioluminescence. The action is pigment screening, and the consequence is reduced exposure. This sequence is documented as a functional interpretation, though effectiveness varies among species and viewing angles. Red tissue, therefore, operates within a larger camouflage system, not as an isolated magic coating. Verified optics establish the mechanism, but ecology determines how much survival value it provides. The next question is whether deep-sea observers perceive the same darkness that humans perceive. Many deep-water animals possess visual pigments tuned to blue wavelengths common in residual sunlight and bioluminescence. To those eyes, a red animal reflecting little blue may deliver a weak visual signal. However, contrast depends on background brightness, distance, movement, and the observer's sensitivity. A dark body crossing faint downwelling light can still form a detectable silhouette. Counter-illumination can erase that silhouette in some species, whereas red pigment alone cannot. From the side, scattering water may reduce contrast more rapidly than it reduces an animal's outline. At close range, even slight blue reflections or body motion may reveal the concealed animal. Thus, Vanishing describes declining chromatic visibility, not complete removal from every visual system. Researchers test reflectance with spectrometers, compare it with measured underwater illumination, and model receptor responses. These methods estimate visibility without claiming access to an animal's subjective experience. Behavioral experiments can strengthen those estimates, but deep-sea species are difficult to maintain naturally. Camera footage adds context, although artificial lights fundamentally change the spectrum being recorded. Red illumination offers another complication because some predators generate or detect unusually long wavelength light. Certain dragonfishes can produce far-red bioluminescence and possess visual adaptations that help detect it. For prey lacking equivalent sensitivity, that private illumination may expose red camouflage at close range. This exception does not overturn spectral filtering. It shows that animals can modify the optical battlefield. Another hypothesis proposes that red pigments sometimes serve physiological roles beyond concealment. Pigments may protect tissues, bind compounds, or arise from diet, depending on the organism. Color alone cannot reveal which selective pressure originally produced a particular red body. Comparative evidence must link habitat, reflectance, predators, development, and fitness before adaptation is established. Even then, multiple functions can coexist within the same tissue. Transparency may outperform redness in open water, but organs, gut contents, and eyes resist becoming transparent. Red or dark pigments can hide those opaque structures while leaving surrounding tissues relatively clear. The cue is incoming blue light. The action is absorption by pigment, and the consequence is weaker reflected contrast. The adaptation works passively, requiring no decision once the animal's tissues develop. Its limits become clearest whenever another light source returns the missing wavelengths. Ecologically, this spectral camouflage helps shape encounters between predators and prey in the twilight zone. A shrimp drifting by day may remain dark beneath the last usable sunlight. At night, vertical migration can carry it upward into broader spectra and different visibility risks. The cue is changing brightness, the action is migration, and the consequence is access to food under altered predation pressure. Red coloration may continue masking the body, but shallower light can reveal more of its hue. Some animals reduce exposure by remaining deeper during daylight and ascending after sunset. Their color, timing and depth together form a layered defense rather than separate tricks. Bioluminescent flashes can abruptly overwrite that defense, tracing nearby bodies with momentary blue light. When threatened, some deep-sea shrimps release luminous secretions that may distract or confuse attackers. The cue is attack. The action is releasing light, and the consequence may be a brief chance to escape. Their dark red bodies can remain inconspicuous beyond the luminous cloud, though outcomes are rarely observed completely. Human exploration repeatedly disrupts these relationships by bringing lamps into naturally dim scenes. Under white light, animals appear theatrical, precisely because exploration restores colors absent from their habitat. If you watch remotely operated vehicle footage, the brightest reds may therefore be observational artifacts. Scientists compensate by measuring ambient spectra and interpreting illuminated imagery with caution. Fishing gear brings red animals upward, where pressure, temperature, and light all change simultaneously. Specimens on deck accurately show pigments, but not their natural appearance in situ. Museum preservation can alter color further, weakening comparisons made without fresh measurements. Conservative interpretation separates observed reflectance from inferred concealment and inferred evolutionary benefit. The human stakes are mainly epistemic. Our lighting can create the very spectacle we try to explain. Future evidence should pair low-light behavioral trials with habitat-specific spectra and realistic visual models. Improved cameras can record blue-dominated scenes without flooding them with broad white illumination. Spectral reflectance measured on living animals would clarify how much surviving light each surface returns. Predator visual sensitivities remain unknown for many deep-sea lineages and require careful physiological study. Field observations could test whether red animals choose backgrounds or orientations that further reduce contrast. Comparisons across depths could reveal whether darker reds track predictable changes in the light field. Genetic studies may identify pigment pathways, but genes alone cannot demonstrate camouflage performance. Experiments must preserve natural pressure and temperature, when physiology or behavior could otherwise change. No measurement will produce one universal disappearance depth, because ocean waters differ continuously. Instead, visibility should be expressed as a relationship among spectrum, distance, background, and observer. The strongest conclusion remains physical. Without red illumination, red surfaces cannot send much red light back. The biological conclusion is narrower, because reduced reflectance usually helps concealment, but never guarantees safety. You could restore the crimson animal instantly with a lamp, proving the color was always present. Turn that lamp away, and the surrounding blue once again drains its visible fire. What looks like vanishing is a partnership between pigment and an ocean that edits sunlight. The scarlet shrimp returns to darkness, concealed not by mystery, but by the measurable loss of red light.